Lance Moore. Malaria is a debilitating and potentially fatal infectious disease caused by the protozoan parasite of the genus Plasmodium. Historical accounts alluding to malaria date back to as early as at least 4,000 years ago, making the parasite one of the most enduring and effective pathogens. Four species of the Plasmodium parasite are responsible for malaria: Plasmodium ovale, Plasmodium vivax, Plasmodium falciparum, and Plasmodium malariae. Of the four species, Plasmodium falciparum is the most pernicious and produces the most fatalities. According to the World Health Organization, in 2015 there were an estimated 214 million cases of malaria worldwide. However, with an uncertainty range of up to 305 million, that number could conceivably be as many as a half a billion cases. Similarly, the estimated 438,000 deaths resulting from the disease might actually be closer to 1 million. 90% of these deaths occurred in sub-Saharan Africa, with children under age 5 disproportionately victims, representing two-thirds of all deaths from malaria. The disease vector for the malaria-causing parasite is the female Anopheles mosquito. The mosquito serves as a host for Plasmodium during part of the parasite's life cycle, with the human victim fulfilling that role for the remainder. The parasite is introduced into the human bloodstream when the mosquito feeds on the blood of the victim. The blood flow current then carries the malarial agent to the liver, where it implants itself within the liver cells and initiates the asexual reproductive process. Over a period of 1 to 2 weeks, during which the human host is asymptomatic, the replication process results in a single-celled organism containing multiple nuclei, which then undergoes multiple cytokinesis, or cell division, to form as many as 64 daughter cells known as merozoites. Rupture of the liver cells then release these infectious merozoites, which reach their target destinations, the erythrocytes, and penetrate them. This process of replication is then repeated and results in large-scale hemolysis, which releases ever more parasitic merozoites to infect additional erythrocytes. The simultaneous lysing of red blood cells occurs every 48 to 72 hours and produces the typical fever and chills symptom of malaria, and obviously reduces the oxygen carrying capacity of the blood. In addition, the rupture of erythrocytes releases toxins which contribute to further debilitation. Some of the parasitic daughter cells differentiate into gametocytes and can be ingested by another Anopheles mosquito if it feeds on the blood of an infected individual. Once inside the body of the mosquito, the male gametes can fertilize the females, and the resulting zygote undergoes numerous nuclear replication and cell division to ultimately produce thousands of new infective cells known as sporozoites. Subsequent migration of the new sporozoites to the salivary glands of the mosquito begins the infection process anew when the mosquito bites another human during a feeding. One interesting evolutionary biological adaptation, or more accurately, a natural selection process favoring a genetic mutation, is the one which altered the structure, and therefore function, of the hemoglobin molecule and resulted in the sickle cell trait. The trait is considerably prevalent among the people and descendants of the inhabitants of areas in which malaria has been historically endemic. The sickle cell trait provides resistance to malaria due to the inability of the Plasmodium parasite to reproduce within the cells, which rupture before replication can take place. A similar resistance is conferred by another genetic mutation which results in abnormal hemoglobin structure and produces a condition known as thalassemia, which also shows greater frequency among those living in or descendants of those living in endemic regions. Both traits, however, must be present in the heterozygous allele form, since homozygosity for the mutant allele can lead to serious debilitation and even death. There is currently no vaccine for malaria, although several prophylactic drugs can be used as a preventative measure. A constant challenge is overcoming the Plasmodium parasite's developing resistance to the chemoprophylaxis.